Now that you have the basic knowledge of what is an antigen and antibody, let's talk about the interaction between the two. As the name suggests, it is chemical reaction between antigen and an antibody produced by the B cells. The antigens and antibodies combine by a process called agglutination and form immune complexes. It is the fundamental reaction in the body by which the body is protected from complex foreign molecules such as pathogens and their chemical toxins. We have already talked about the specificity of antigen due to the conformation of epitope. The antigen-antibody have complementary fit. If the structure of the epitope is not a complement to the shape of paratope, that the complex is not formed. The circulating antibodies binds to specific antigen with high affinity to form antigen-antibody complex. The immune complex is then transported to cellular systems where it can be destroyed or deactivated. The antigen-antibody interactions can be classified into primary and secondary interactions. We will go through these two types of interactions one by one. Starting with the primary interactions. Binding depends on a close three-dimensional fit, allowing weak intermolecular forces to overcome the normal repulsion between molecules. Although binding between antigen and antibody involves only non-covalent forces and therefore is theoretically reversible, in practice the high affinity of most antibodies means that they rarely become detached from their targets before these are destroyed. The weak and non-covalent interactions such as van der Waals forces, hydrogen bonds, electrostatic interactions, and hydrophobic interactions are responsible for binding. As these are weak forces of attraction, antigen and antibody must come in proximity in order to bind. The two properties of an antibody, that is affinity and avidity, also play a major role in the antigen-antibody interaction. Moving forward, to understand it more easily, we are going to break down the primary interaction into two headings: affinity and avidity. Let's talk about affinity first. We have used this word multiple times already, but what does it mean in case of antigen-antibody interaction? Affinity of an antibody means the strength with which one paratope binds to the epitope. In simple words, it is the force of attraction between the paratope region on the antibody and epitope region on an antigen. The force of attraction is directly proportional to the complementary fitting of the epitope, meaning when the shape of the epitope is a perfect fit, more bonds are formed resulting in strong binding. Whereas if the shape of the epitope is not complementary to the paratope, less bonds are formed resulting in weak binding. Avidity of an antibody means the total strength with which an antibody binds to epitopes. It involves all the antigen binding site of an antibody. If you recall, we talked about different immunoglobulins in the previous section. That concept will now come in handy to better understand avidity. As you know, IgG is a monomer and has two antigen binding sites. HEAA is found in monomer, dimer, and trimer forms. So it has 24 and 6 antigen binding sites respectively. IgM has maximum of 10 binding sites. As the number of antigen binding sites increases, the strength of overall binding also increases, making the antigen-antibody complex more stable. Another concept that you need to familiarize yourself with is the cross-reactivity. It is defined as the ability of an antibody to react with similar epitopes on different antigens. Cross-reactivity only happens in situations where similar epitopes are present on different antigens. That was the primary interaction. The secondary interaction starts after the formation of antigen-antibody or the immune complex. The characteristic features and the function of an immune complex is to initiate an immune response to fight off foreign invaders or pathogens. One way of doing that is by stimulating the complementary system. If you have seen our video on the complementary system, you should recall we talked about the different pathways through which it gets activated. The classical pathway is that one that also known antibody dependent. The first complement, that is C1, is made up of 3 distinct subunits, C1q, C1r, and C1s. C1q binds to the effic portion of complexed antibody, possibly under the influence of a conformational change in the shape of the Ig molecule. Activation of the classic complement pathway follows. You can check out the video for a more detailed look at the classical pathway of complementary system. That is all about the antigen-antibody interactions. For more immunology videos like this, stay tuned to sqadia.com.